Welcome everyone. I'm Bill Daniel, the Union County Prosecutor, and it's my pleasure to introduce you to the world of science and justice inside Union County's very own forensic laboratory. Today, through this virtual tour, you'll get a glimpse behind the scenes and witness firsthand the crucial role science plays in upholding the law and seeking justice for the people of Union County. But before we step into the lab, let's rewind a bit. In 1972, Union County made history by establishing the first county prosecutor's lab in the entire state of New Jersey, testing for controlled dangerous substances, and made history again in 2008 by establishing the first county DNA laboratory. Since then, we've become a leader in forensic analysis and demonstrated our commitment to quality by earning prestigious accreditation from the American National Standard Institute's National Accreditation Board, through strict compliance with international standards and the Federal Bureau of Investigation's quality assurance standards. The heart of our lab beats in its technical unit, housing two vital sections: the Controlled Dangerous Substances section and the Forensic Biology section. Now, the CDS team works tirelessly to analyze illegal drugs, providing vital evidence for narcotics investigations and prosecutions. Their expertise is so trusted that the state of New Jersey recognizes their analysis as courtroom worthy, often eliminating the need for expert testimony. Meanwhile, the Forensic Biology section delves into the realm of DNA, identifying bodily fluids and generating DNA profiles crucial for legal proceedings and even solving cold cases. These dedicated forensic scientists are the silent heroes behind the countless solved crimes and brought to justice criminals. Every year, our lab tackles thousands of cases, each playing a significant role in ensuring public safety and bringing closure to victims and their families. From routine drug analysis to complex DNA profiling, our scientists leave no stone unturned in their relentless pursuit of truth and justice. So, without further ado, let's step into the virtual world of the Union County Forensic Lab and witness the power of science at work. Get ready to be amazed, informed, and perhaps even a little surprised by the unseen forces that help keep our community safe. This tour promises to be an eye-opening journey. Are you ready? I know I'm ready. I'm excited to have you here. My staff is ready. Let's get inside and get started.
When evidence comes into the laboratory, it is received by our evidence receiving staff, where a chain of custody for the laboratory is started. The evidence is then put into our secure vault, and it remains there until testing starts. What type of testing depends on what the evidence is and what type of testing is requested. The types of evidence can vary widely. We get everything from swabs, standardized things like sexual assault kits, clothing, weapons, to the unusual things. Sometimes we will get, very large items. We've had portions of flooring, we've had portions of cars, we've had a mannequin come in. The drug evidence tends to be a little bit more straightforward. Evidence that is collected that's believed to be an illegal substance. So our job is to test it and determine exactly what is in there, and if it is a scheduled substance, what that schedule is.
So now we're going to start with the drug vault, and because we house controlled substances there, we have to meet certain security requirements. You'll notice that this has a big giant door with a combination lock, and that's as well as our security system in the outer portion of the vaults. And this is because the DA has specific regulations on where and how you can store drugs. So in addition to the giant door, which is closed at night, we actually also have a day gate. So each vault has a badge access, and that only analysts have access to the evidence. We have a separate section for incoming evidence, so this is the area where we have not tested the evidence yet. We are lucky that it's not overflowing at the moment. In these shelves, we have the cases that we have already tested. Some are smaller cases, some are larger ones, as you can see. We have to store the evidence for the typical time frame is 5 years after the conclusion of the case. So if there was any sentencing after conviction, it has to be 5 years after the conclusion of the sentence. There are a few exceptions where we can get special approval to destroy them after a year and a day. That's usually for really large cases where we don't have room to store them. And in that instance, we'd have certain documentation and we'd have to keep the portion that we tested. Anything really can have drugs on it, apparently. Anything can be used to take drugs. We had an apple that was used for smoking. Obviously, we don't test food products with suspected marijuana anymore, but we used to get a lot of like cookies, brownies, even chips. I had Cheetos, which did not test well. So now we are in the biology section hall, and evidence here is a little more somber because it's often tied to more serious crimes. You'll see we have a large wall of sexual assault kits. One of the projects that we're currently working on is making sure every single sexual assault kit that was taken and reported is tested, even if it's not going to be used for prosecuting a case. This is a huge undertaking and it will take many years, but we're really working through getting that backlog tested. We also hold Jane or John Doe kits, and those are kits where no crime was reported, no one has consented to testing, so we are not allowed to test those, but we keep them for up to 20 years after the crime, just in case someone wants to change their mind. Even though those DNA profiles might not be used for prosecuting a case, they can be put into CODIS and possibly hit on other crimes. So we think that it's really important to get everything tested, even if it takes us a few years.
This is one of 2 examination rooms that we have here in the biology section. The purpose is to retrieve evidence that has come in from the law enforcement, and we retrieve it from the vaults, and we'll bring it in, we'll open it up onto a clean surface such as this, where we've bleached the surface, we put down a sterile paper, and then we open up the item. We will document what the item looks like, if we see any visible stains, and then we may test some stains if we see them. On this particular item, which is a shirt, one of our jobs is to try to identify if it might be blood. And that would involve taking a cutting of a stained area and then applying chemicals for a quick, what we call a color test. This color test is presumptive in nature, meaning that if you get a positive result, that it does not say it's blood, it may just indicate that it's blood. If we do have a positive result, then we can take another cutting and use that for a DNA test to determine whose blood it might be. You can see that there was a very quick, bright pink color, indicating again that there is blood present on the shirt. So this tube will be put into one of our tube racks and then brought down to the other parts of the laboratory where the DNA is done. Once they are in the DNA analysis areas, those samples will never go back to an examination room. It's always a one-way flow of our analysis, and that is to prevent any kind of contamination that can occur in a laboratory. We are taking every steps, all the steps that we can take, including this one-way flow. So this poster represents DNA analysis. Basically, the first part would be, again, identifying possible biological material, such as possible blood on this t-shirt, taking a cutting and then subjecting it to the 4-step process of a DNA analysis. The first step, we're looking to get DNA from the inside of cells. In blood, that's white blood cells. So the DNA is housed within a structure called the nucleus within a cell, and basically we need to break open the cell, break open the nucleus, and then get to the DNA. The DNA has to be extracted or isolated from the stain, and then it has to be purified from all the other components that are present. So that's extraction. After that, we need to quantitate our sample, which means to determine how much DNA is present. In this particular step, we find out if it's a human DNA versus not human DNA. We also find out if there's male DNA present, and if there is even enough to go forward. Some samples are just they have just a very low amount of biological material, and therefore we cannot even work with them. And some have very large amounts, so we need to know that as well. After that second step of quantitation, we perform a third step called amplification. on instruments called thermal cyclers. And these instruments help us to target areas within our DNA that we all have, but have found to be different because of just genetics. You have a different mom and a different dad, and the random association between the egg and sperm. And so we target actually 24 areas that are different between people, and we will make lots of copies of those areas. We make the copies because, again, most of our samples are not of the best quality or quantity, and therefore we need enough DNA to be able to get a result. During the amplification step, the DNA is targeted and copied, and it's also tagged with fluorescent dyes. These fluorescent dyes help us to visualize DNA. This shows a complete DNA profile here, and you can see that the areas that we target are in these green rectangles at the top of each of the colors. And those areas that we target are collectively called loci. They have different names, but basically none of them make you look a certain way. They all have internal functions, but nothing external, again, to how you would look. The DNA profiles, the DNA peaks, which are here, for example, are labeled by software that we use to analyze the data. And for example, these first two are called a 15 and a 16. And then we have a 14 and a 16, and so on. The complete profile, again, is a picture of a person or an evidence item. And if you just looked at just the blue results, for example, we have a sort of example comparison here, where these blue results are for the evidence sample, which could be the blood stain on the shirt, and then the ones below would be the blue results for the suspect's reference. And you'll notice that those numbers are different. So we can exclude or say that the DNA on the t-shirt, the blood stain on the t-shirt, the evidence sample, could not have come from the suspect, because if they did, they would be the same. Sometimes we have an evidence sample result and we don't have a suspect's reference. Well, in those situations, we are a CODIS participating laboratory. CODIS is the Combined DNA Index System. This is a database which involves many different types of DNA profiles, to include those of convicted offenders and arrestees in New Jersey of certain crimes. And we can put this profile electronically into CODIS and have it searched against convicted offenders and arrestees for New Jersey, as well as for the rest of the country. DNA has made a lot of advancements since it was first used in casework back in the 1980s. One of the major advancements is the size of the sample that you need. Back then, in the 80s and 90s, you needed, say, a quarter-size stain of blood, probably equal amounts of semen. And even you can see on this shirt, any one of these blood stains is much smaller than a quarter, basically. But even the smallest stain that you can see, and even some that you cannot see, would be enough for a test to work and generate a DNA profile today. Prior to DNA testing, the only testing that could be done on a shirt like this would be to determine if there was blood or not, and maybe if it was human or not, but nothing else. So we routinely will go back with the cold case unit of the Homicide Task Force and go through cases that we might be able to test now, given our technology, but could not have been tested at the time that the crime occurred.
In this section, this is a controlled dangerous substance section, where we process all the evidence to be tested for the presence of controlled substances / illicit substances, which just means that state and federally controlled Schedule 1 through Schedule 5. A Schedule 1 substance is something like heroin that has a high potential for abuse with no medical use. A Schedule 2 substance is something like fentanyl, where it has a limited medical use, but also a high potential for abuse. All the way through Schedule 5, which has medical use with low potential for abuse. When I started like 17, 18 years ago, it was mostly plant material, marijuana, cocaine, and heroin. And around 2016, 2017, we started seeing an uptick in fentanyl submissions. To the most current, I'd say most of our drug submissions are fentanyl and or opiate submissions, methamphetamine, and cocaine. As far as opiate submissions, I would say just about all of them contain fentanyl and heroin, or just fentanyl with some kind of fentanyl analog in there as well. We do get clandestine made pills that contain fentanyl, such as they're being marketed to the public, stamped like they're an oxycodone pill that you would get from your local pharmacy. And when we go to test them, we actually find out they contain fentanyl. During the normal course of a business day, we test several different substances that could lead to a chemical spill or some kind of possible overdose of the fentanyls that we're seeing. So to be prepared for that, we have several stations set up to clean up spills, and also we have Narcan on hand that would, in case if anybody had a fentanyl overdose, which fortunately has never happened here, but we always have to be prepared that it could. Right now I'm handling a submission of about 250 pills that we're suspecting to be methamphetamine. It's usually what we see in these. These happen to be just round and orange, but we do see various shapes and sizes in just about every color you can imagine. Sometimes they turn out to be other things. So that's why we're going to do our testing to see what we can confirm this. So one test that we could use when we get an unknown powder is we would do a presentive screening test known as a color test. And in this case, we would add some of our reagent to a clean spot plate to make sure it wasn't contaminated first, and then we add a small amount of our sample and we note any color change. In this case, it turned purple, which is an indication of an opiate being present. We then come over here and add the appropriate solvent, which is then going to be injected on our gas chromatograph mass selective detector, also known as GCMSD. How it works is you have your sample dissolved in a solvent and placed into a vial, which it then gets injected into the instrument. It flows through the gas chromatograph part, which is the long column. It gives it plenty of time to separate any components in this sample. It then flows into a detector, which is bombarded with electrons, which causes the sample to fragment into a specific ion pattern that is specific for that drug. We then compare that to a reference material for identification. If the GCMSD doesn't give us enough information to make a confirmation on a drug, another instrument we use to make our drug confirmations is called an FTIR. This one happens to be powered by liquid nitrogen, which gets put into the instrument. Here we have our sample on a salt plate where we focus in with the microscope. In this case, we have methamphetamine. We then compare it to a reference material that was ran on the same instrument for identification. Once we finish our analysis, which I just took you through all the various tests we do, we then put our evidence back, permanently sealed into the drug vault. After that, we review all of our data. We come up with our conclusions and our findings. We prepare a report, which then is reviewed by 2 other chemists in our section. Once that's done, the report is published. We then may be called at a later date to provide expert witness testimony.
Now that you've seen what the lab has to offer, we can talk a little bit about what it takes to work in this field. I have a background, my undergraduate degree is in biochemistry and my graduate degree is in forensic science. As a laboratory, we tend to look for people who have backgrounds in either one of the natural sciences, biology, chemistry, or physics, or who've taken specific forensic science courses. We are looking for people who've had a lot of hands-on experience within their laboratory courses. So some forensic science programs will lean more towards pattern disciplines, things like crime scene investigation, trace evidence, and those are good courses to seek out if those are the disciplines you're interested in going into. If you're interested in working in a laboratory, you should try and look for programs that have a very heavy lab focus, where you're going to be getting hands-on experience with instrumentation and getting used to doing laboratory protocols. You also, if you have the opportunity to do research, that's a great way of getting additional hands-on experience, or even internships at laboratories. We offer them here for a limited number of students, and we are looking for people who have some sort of laboratory hands-on experience because we use them to do that kind of work here. For people who are interested in a career in forensic biology, specifically DNA, there are specific courses that you need to make sure that you take in college, or we cannot consider you for a DNA analyst position. And specifically, those are molecular biology, genetics, biochemistry, and statistics. At this point, especially for forensic DNA, you're probably going to need a master's degree for an entry-level position. Some of the other disciplines, it's not as common, but absolutely forensic DNA. There is a lot of interest in that field, and the large majority of your hiring pool are going to have master's degrees. CODIS was started in the late 90s, but of course at that time was very small. As time has gone by, it's grown. There's now several million profiles in CODIS, and that increases the likelihood that there's going to be some information generated. Even if it is not a link to a known individual, having multiple cases that can now be linked when they were not previously linked can help progress the investigation and lead to a resolution. What we've seen starting in about 2017 that has continued to build year upon year is we are seeing a lot more mixtures of samples, and we've seen a significant increase in fentanyls. The large majority of glassines that we test now are mixtures of 3, 4, 5 Schedule I substances, mostly mixtures of different types of fentanyl, but we're also seeing mixtures of different types of drugs together, which can be really dangerous from a perspective of someone who's using it. If you've developed a tolerance to fentanyl and now that fentanyl is mixed with methamphetamine, that is not something that you've built a tolerance to. And we have seen those sorts of drugs that are mixed together. At this point, it is much more common that we see a mixture of drugs than we see something by itself. So one of the things that's unusual about forensic science is that we are having to go and explain our results to lay people. Most people get into science because we don't like talking to people. We like being in the lab, we like doing all of the bench work, we don't necessarily like having to present that to people. But forensic science is this very kind of unusual place where not only do we have to present it to a bunch of lay people, we have to make sure they understand it. And again, they understand the limitations and what the testing can do and what it means. And we need to do that in a way that's relatable, because you're talking to a group that have very diverse backgrounds, very diverse education levels, and the stuff that we do is really complex. It's not easy to understand. And we need to make them understand it because the consequences of it are big. You know, these results are part of the overall judicial process, and we need to make sure that the jury can properly weigh those. So one of the things I like to tell people who are getting into it is, please be aware that you are going to be going to testify. The amount is going to vary. In general, our DNA analysts testify much more frequently than our chemists do. But everybody here has been to testify at least a couple times. And it is something that you can work on. If you hate public speaking right now, that doesn't mean that you're never going to be good at it. Like any other skill, it's a skill that can be developed. And if you know that you want to go into forensics and that this is something that you want to do, start taking public speaking classes, join an improv group, get involved with performing arts, anything that's going to get you in front of people and get used to talking. It's not that you can't do it on the job. That's how I learned how to talk in front of people. But it definitely makes it easier if you have some level of comfort beforehand, because then you don't have to worry about the nervous jitters part of it. You just have to worry about the conveying the complex scientific information part of it. Just for kind of clarification, all cases or all evidence items that were seen during the course of this tour were not active cases, and we did not compromise any evidence or any active cases in the filming of this tour.